Class we have talked about this thing, that is what is database, and in database we have seen various kind of database, for example primary database, secondary database, composite database. Also based according to bioinformatics, we have seen in case of primary we have seen sequence database, structured database and functional database also. In case of sequence database, we have further classified into primary and secondary database, and we have seen in case of primary sequence database, three common database were there. One is NCBI, another one is EMBL, and third one is GDBJ. In this case, we have studied our first experiment, that is exploration of this database, that is about NCBI. We have done that. I want to add little bit that in case of sequence, we can also submit the data. In the last experiment, you have just retrieve your data in terms of FASTA format and GenBank format, but there are two different tools that can be used for the submission of data. These are Bankit as well as Sequin. As the name indicates, we are inputting something in the bank, in the GenBank. So if the sequence length is quite small, if you are talking about the protein sequence or the small gene sequence or the exome thing, you can use this Bankit kind of thing. It is an online tool which is available. You can go to NCBI, make an account, and in this Bankit thing, you can add the information of your gene or the exome region. But if you are talking about the genome sequence, or you have sequence by the use of NGS or next generation sequencing, or you want to add bigger data, then you can use Sequin. It is an offline tool which can be used for the submission of your nucleotide data or sometimes protein data also. Majorly Sequin is useful for the genomic data also. So I'm not demonstrating this experiment for Sequin. Otherwise, you can go and simply download this Sequin on your system, add some information, add some random nucleotide ATGC in a combination, and submit that thing, and it will automatically appear in the in your NCBI account. I think in the last class I have talked to you about three different things. One was version number, one is GI number, that is gene bank number, and other one is accession number. So we can say that it's a primary, secondary, or it's a composite, or it may consist of the various version or the various modification of this kind of thing. So we have discussed this thing in the last class. So we are going towards the statistics part right now. After that, I will show you different kind of formats other than FASTA format, which are quite useful in case of bioinformatics. As the stats shows you, due to COVID, this thing, the amount of data which is generated by the laboratories is quite huge, and it is going in an exponential way. And due to vast amount of data, we need database. And which format which is commonly used in case of this is nothing but FASTA and GenBank format. So we can say that FASTA consists of only sequence information, while in case of GenBank, two different things are there. One is features and other in sequence. In case of feature, it talks to you about the locus number, accession number, version number, number of publications, all the things are there, CDS, exon region. But in case of sequence, it is arranged in the form of 1 to 60 kind of format, where it is further fragmented in 10 base pair each, and at last it is this, while in case of this it is there. So the common difference is in terms of sequence arrangement, and second is about the processing. It is very shorter format and it takes very less space, and due to this its processing time is quite huge or quite fast. So while in case of GenBank format, it is quite larger in terms of information. It consists of lots of information, but it takes lots of time for the processing and takes lots of space, and due to this it is less used as compared to FASTA format. Whatever tool we are using or we will use in bioinformatics, we will majorly use this format, that is known as FASTA format. So other than that, this FASTA and GenBank format, there are various other formats that are available that are most commonly used. These are, for example, I'm showing you. First one was FASTA and GenBank. Other than that is EMBL. Have you heard about this database, European Molecular Biology Act? Last class I have shown you a simple figure, and I have illustrated you a hypothetical story. For example, during Human Genome Project, US has contributed for the nucleotide part, DNA and RNA, and they made the database known as NCBI, National Center for Biotechnology Information. After that, European countries say that they have taken the nucleotide, DNA and RNA, but should we work on? We should work on the protein thing, and they make a database that is known as EMBL, and it is also known as EBI. It does not mean it does not consist of any nucleotide sequence. It consists because they exchange the information, but it is primarily useful for the protein database. So the second database was EMBL, and they made, or you can say they have just copied the format of GenBank. In GenBank, locus number is there, accession number is there. They have just changed. They have changed it as ID number, and they have changed it as version number. BI they have used, for example, for sequence SQ they have used rather than returning writing sequence. So they have used short form of that of GenBank, and they have modified that format and made them as EMBL format or European Molecular Biology Lab format. Other formats are PIR format. In the last class, I have told you that in case of FASTA format, it starts with a greater than sign. But in case of PR, it also starts with a greater than sign, but it uses this semicolon kind of thing. It is similar to that of FASTA, but it is majorly used for proteins. So other format is GCG format, which is also known as Genetic Computing Group. The full form of PIR is Protein Information Resource, and it was developed by university. And GCG format, it is primarily used for DNA sequence or nucleotide sequence. It is almost a copy of this GenBank. In case of GenBank, the sequence arrangement is like this. So in single line, we are getting 1 to 60 base pair, but in case of GCG, we are getting 1 to 50 base pair. Also, fragmentation of this particular segment is again of 10 base pair. It means 5 fragments are there in a single line. Along with that, it gives you the composition of each base pair. How many number of A's are there? How many number of T, G, and C are there? But in case of this GenBank, it is not available. This kind of information is not available. Base composition is not given. The other format is ICF format, that is Intelligent Genetic Sequence format, which is also, you can say, a copy of this or the refinement of this PIR format. I'm showing you the example of this. So these are the most common formats that are used in bioinformatics. The other format which is useful is MSF format or multiple sequence format, in which we are using 2 or more same kind of sequence information or the sequence format information. For example, if I am writing in a team, there are 6 people. Someone is working on human, another is working on mouse, another one is working on rat. So if I am taking 2 or more sequence of same type of format, then it is nothing but the MSF format or multiple sequence format. Or we can say that if 2 or more people are working on GenBank format, then this particular format becomes MSF format. Are you getting my point? So this is nothing but the MSF format. So this format you are most familiar about. This is FASTA format, which starts with this thing. Sometimes an asterisk mark is also added at the last. It means it's an reference sequence, or this sequence has been patented. Or if you want to access this particular FASTA sequence, you need permissions. So sometimes it is there in some sequence. Otherwise, the genes which I have sent to you, the accession number which I have provided, all are free of cost. So it is so there is no asterisk kind of thing is there in case of FASTA format. Other than that, I have told you regarding this thing 1 to 60 in the last class also, and the identifier is also there. This is nothing but the GenBank that is available at NCBI. Other format is EMBL. For example, you can see here it is locus number, base composition is there, type of the sequence, linear or the date of submission. All the things are there. For example, here it is, instead of accession number, it is ID number. It is showing which kind of standard RNA. Here it is, there it is linear. It is the primate. It is the base composition. Again, accession number here it is given. Or also origin of the species. Under that it is taxonomy. It is and here it is sequence. For example, you can see here it is there sequence, and in this case here it is SQ, nothing but sequence. All the things are given. Are you getting my point? In this case also it is just nothing but the copy of the previous version. Also the other one is known as NVRF format. Is it is a university which has made this particular format, that is protein information resource. It is similar to that of FASTA format, but some addition has been taken place. So here it is. No, you can add some information, as in case of FASTA, we have seen that there is an optional condition that you can add information regarding gene sequence or the type of organism. Otherwise, it is majorly used for the protein sequence also only. So here it is only protein sequence, but we can use also for the DNA sequence also. But here it is given for the protein. So it is similar to FASTA format, or you can say that the common fundamental formats are nothing but GenBank or FASTA format. Other formats are just a you can say modification of the previously existing format, or you can say or the copy of that. Other one is GCG format, which I have told you 1 to 60, 1 to 50 is there, and the here it is not given about the in this screenshot, where A, T, G, C number is also given in that particular thing. So you can check that. Other is IntelliGenetic sequence format or IG format. It starts with this. Some information is there, but no greater than symbol is this. I have told you it is a modification of this PIR format. So these are the ones. You do not have to remember all the things, but you have to, yeah, you have to able to recognize some common formats such as FASTA format, such as MSF format, or you can say GenBank format, which is quite useful in your bioinformatics. And this kind of format is quite useful in multiple sequence alignment, where you want to find out the conserved region or the common region that is existing among all the species, or semi-conserved region that is available among all the species, or unconserved region. All the things we have to find out by the use of this format that is known as multiple sequence format. Here, which format is used 2 or more times? Can anyone tell me? Here only sequence information is given. Along with that, the name of the species is given. So it is nothing but the FASTA format. Here we have used 2 or more FASTA sequence here, then it becomes a multiple sequence format. Okay. So it is quite useful for finding out multiple sequence alignment. We will discuss this thing in the 6th experiment. But you should remember these formats are useful in bioinformatics for doing different kind of experiments. So one thing regarding the PDB format, it will be helpful in our 9th and 10th experiment. And for example, another is H group. For example, if I'm talking about glycine in this case, it is H. So in this case, the first atom should be nitrogen. Here it is 1, here it is 2. So 1 is nothing but the nitrogen. And which amino acid it is? Because there 2 carbon is there. So in case of acid, extra COH should be there in case of R group. So in this case, the first, for example, I'm saying it is N. Next is alpha carbon. This alpha carbon is represented by Ca. Next, for example, next one is for carboxylic C. Third element is there. Fourth is oxygen. This is fourth one. Fifth one is also something group is there which is attached to this. This whole things is available in the form of 3-dimensional coordinates. These 3-dimensional coordinates represent this kind of format that is nothing but the PDB format. I think in the last class I have also told you regarding this sequence and structured database, and under sequence and structured database I have told you regarding 2 types of primary and secondary database. For example, if you add some raw information from experimental result, where you add in fundamental database either NCBI, EMBL or DDBJ, you are submitting something and they are checking something and giving you an accession number. After that, this information is further processed or further refined and it becomes the protein sequence information. If it is DNA, then it becomes protein, or if it is protein, then it is refined sequence information, which comes under TREMBL. After that, it goes further to the structured thing. This is DNA sequence. This is protein sequence. Further refined, we are again refining this thing in the form of primary structural information, which comes under either CAT, SCOPE. These are the database which are there. After that, further information is processed and it becomes present in PDB database, or which is also known as RCSD, Research Collaborative Structural Bioinformatics. And the format of this particular database is nothing but PDB. And this is the purest form of the data. Or I think in the last class, I have also told you this pyramid kind of thing. The lowest one is known as data. If we pick only relevant thing out of data, then it becomes information. And we are further filtering this thing, it becomes knowledge. And if we are further filtering it, it becomes intellectual. This is the topmost level or the quaternary level. And here it is. PDB comes under this 4th level. What is the primary level? It is nothing but the raw, or you can say is the data or the primary sequence database. Are you getting my point? All the database. We have discussed all kind of formats, but this is a tool which I am talking about, Sequin. You can download in your system. You can try in your system. Either you can submit in the GenBank or in the EMBL. If it is in email, it should probably ask for the proteins. If it is in the GenBank, it will ask for the nucleotide. You can give any information, any random information, and it will submit your data in the NCBI. If it is submitting your data, it will allocate certain number. In 2 years before, it was giving you accession number, but right now it will only give you the application number where you are submitting. So it can be useful for that. Before proceeding towards this thing, you should have to first make your account in NCBI, and it can be easily made. You can do that. So this experiment we have done in the last class,